So, forensic palynology, it takes the basics of palynology as a whole, which is the study of pollen and other spores, and this is used to prove or disprove relationships between people and crime scenes. So, it can cover any type of cases such as homicide, genocide, forgery, drug trafficking, and rape. This is, however, the most underused technique within forensics, just due to the over reliance on our other techniques that we use, simple ignorance on just exactly what this field is. There is also a lack of experts within the field, so it does make it a little bit hard for us to find reliable sources. And because it's not really used all the time, there is just a lack of standardization that gets in the way. And like to mention before, this application is solely dependent on the amount and type of pollen that was collected at a crime scene, as well as the characteristics of the region that we're questioning. All the characteristics of a pollen spore, we can basically kind of compare it to a fingerprint. They all kind of have their own very unique traits that we can go off of. So there are four major applications of forensic palynology. This is to link suspects and evidence to crime scenes, estimate the season of death or time of exposure of a body. We can also narrow down the search to locate a gravesite or a body based off of the pollen, and we can even confirm or disprove alibis of the suspects, basically just confirming if they are where they said they were based on the pollen that we found. The methods that we use to analyze this are going to be light microscopy, electron microscopy, Raman spectroscopy / stable isotope analysis, DNA barcoding, and orthogonal analysis. So light microscopy was mainly used around the 1930s to 1950. It is the oldest method of palynology. It is able to count and identify pollen from the collections that we get. This is done by hand lenses at the scenes that will take pictures. We then blow these up and see how they react to light, and then we recreate those drawings, and that's how they're studied. Electron microscopy got a little bit more famous in 1986. Instead of using photons to create the image, it uses a beam of electrons, and that's how we're able to visualize the structure and composition of the pollen. So this is a little bit more specific. It is able to see the morphological comparisons and the taxonomy of the species we collected. Raman microscopy and DNA barcoding were around the same time and used interchangeably from 2000 to 2018. It's a combination of chemical analysis and traditional light microscopy. So not only can it do the morphological comparisons and taxonomy, because the precision of the identification is so sensitive, we're able to actually identify the members of within the same taxonomy. And then DNA barcoding, as its use among science, was able to just target specific markers in the genome. And specifically with pollen, if we take a look at the plastid genome, this has a high variability among species. So being able to identify it really closed in on what species we were looking at. Orthogonal analysis is around 2019 to the present. It's basically just a combination of independent analytical methods. So it's a mixture of all the ones mentioned above, and it's just to increase the reliability of the analysis. So the case that demonstrates the first use of palynology is the murder in Austria of 1959. It happened in Vienna when a passenger on a boat along the Danube River went missing. He was accompanied by a coworker on the boat, but that coworker claimed that he did not know what had happened to him. And since there was no body, no physical evidence, or solid suspects, it kind of ended up at a stalemate. Eventually, though, when they were searching the coworker's cabin, they discovered that he had muddy boots there, and on one of the shoes, there did appear to be pollen grains on them. So it went to the most famous paleontologist at the time named William Claus, and he was able to use light microscopy at the time, and he was able to determine that the pollen on the boot was a specific type of grain called Cara, and this grain was eroded from a sediment of the Miocene age. So he knew this was a very rare grain, and he knew that the only area in Austria where this grain had inhabited was a hickory tree near the Danube River. So when they pressed the questions to the coworker about the grain of pollen that they had on there, he eventually had confessed and led them to the hickory tree that William Claus had found, and that was the body dump site.